Okay, so we have a very exciting session ahead of us right now. We are going to be talking about data management and sharing, which is a passionate topic for all of us, I know. So thank you for coming back from lunch. Thank you for those of you that are joining us again virtually. And I will hand it over to Caterina Strambio-De-Castillia. Oh, sorry, Judith. Judith is going to do a quick introduction for this session first and introduce the speakers. Thanks.
Hello, everybody. Thank you for choosing to attend this session instead of enjoying the swimming pool. But I swear we're going to convince you that managing and sharing data can be very refreshing. Okay. All right. So I'm Judith Lacoste. We met earlier. So we're about to start a subject that is very close to my heart. Managing and sharing data, and I told you earlier that our goal is to automate open, reproducible, and fair science and have this done in line with the bioimaging community and standards. Now, this subject can be defined as complex, challenging, difficult, scary, time-consuming, not rewarding, long-term, underfunded, and impossible. And yeah, these are all terms that have been presented to me in the past year. So we want to make use of the a car analogy and trying to make this more accessible, more easier, rewarding, encouraging, and especially more fun. So I'm self-deprecating here to show you what I do when I'm not doing microscopy, and I do car mechanic. So this is me upgrading the suspension of a pickup truck. All right, so let's go into the mechanics of managing and sharing data. This is just a slide that I find we came up with this idea recently, and here's what's happening. Our first speaker is Josh, and I'll give a more proper intro to Josh, but really Josh is going to tell us about the car system, what's under the hood that we're not seeing when we operate a microscope, what we're not seeing when we're dealing necessarily with all the managing and day-to-day operation of our core facility. Then Caterina is going to get on board, and she's going to present us her work, which is really dealing with the car dashboard, really interfacing between the car system and the driver. And our last speaker is going to be Caroline Miller, who is really our driver today. And she has a very interesting twist because she's been really active in the biopharma industry. So that will also be a very useful perspective to find. So without further ado, I would like to introduce formally our colleague, Josh Moore. Josh is online with us. And Josh is a research software engineer, and really he spends his time working to build reusable software infrastructure for us, the bioimaging community, to be able to achieve our goals about open, reproducible, and fair science. All right. So Josh is going to talk about the work he's been doing for several years. He's heavily involved with open microscopy environment. He's also part of German Bioimaging and FDI for Bioimage, as well as the Quarepidemie organization. All right, Josh, the floor is yours.
Thanks, Judith. I think I should be audible. Could someone give me confirmation just in case?
Yes.
Thank you. Awesome. Hi, everyone. Yeah, so Judith maybe said most of the things I was going to say for my introduction. She was a bit kind, saying several years and not many, 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 many years. I tend to use the beginning of all of my presentations to introduce handles and say, as a community, we probably need to move off of Twitter. So I'll give you also links to Mastodon. That's where I'm trying to establish a scientific community to discuss. If you are moving to another community, I guess, let me know and I will join that as well. So I think it's really important that we have a place that we can discuss things, and I'll actually end with another place we can discuss things. As Judith said, I'm affiliated with German Bioimaging. So there I'm a research software engineer, and as she said, I build infrastructure. German Bioimaging is a nonprofit scientific society, so representing the imaging core facilities in Germany. And so I guess from all of us, I guess to Mexican bioimaging community, congratulations on the signing. Very much looking forward to bridging communities and talking more. I really wish I was there. It would make this so much easier, but nevertheless, and I am a part of OME, which is some 20-year-old infrastructure project building software to do all these things to try to make research data management easy. So that's what I'll really be talking about and representing. Because as Judith said, research data management can be complex, difficult, et cetera, et cetera. It's intended to get us to the point where we have our data being fair, so findable, accessible, interoperable, reusable. But lowercase fair, it is not, meaning it's a burden that gets put on all of you. So we're each asked to do this. You know, the NIH white paper came out, we're being asked to do it more and more, but we don't necessarily have all the tools ready to share data as we need to. So that's what I would really like to get across is there are things out there that you can use. I think we're going to have lots of different metaphors about, so we have the car metaphor, we have, you know, Clara here, who's pushing her data, trying to share it, and you'll see some more metaphors in just a second. So we're really trying to get our head around how it is that we move data around and do it in the best way possible. For what it's worth, this image is available CC BY. It was done by an illustrator for a chapter on volume EM data. And everything I show will be CC BY, and you're welcome to use it to convince people to try to get their data out and have it shared and open. So I assume Vanessa and Nikki and others will share the slides with you, and you're welcome to do what you'd like. So one of the reasons that sharing bioimaging data is so difficult is and it makes us special, is this complex, high-dimensional acquisition of data. So what's coming off the acquisition systems is certainly in the OME world, we consider at least 5-dimensional. But then you can have, of course, here, if I use my mouse, you know, you can have many images that are in a well, many wells on a plate, many plates in a screen. And before you know it, you have millions of images that you somehow have to deal with and put somewhere. And as users, you and your users rightly expect to be able to access all that data interactively. You know, you basically want a YouTube mixed with a Google Maps availability of all the data instantly. And I can tell you from the research software engineering point of view, from the back end in IT, it is simply a difficult problem. You know, Google has tens of thousands, if not hundreds of thousands of, I don't even know, developers working on these kinds of problems. And we as a community really need to come to grips with how we're going to do this. So if you are struggling to do these things, there's a good reason, and that's fine. That's what we're going to talk about. So what I would really like to offer you is kind of some turnkey solution that, you know, there's this thing and you go to a website and you download it and it just works. And all the imaging coming off of your acquisition systems is instantly stored and is accessible from all available tools, but we're pretty far away from that. So this is, again, a different metaphor that we came up for the European Light Microscopy Initiative meeting this summer, talking about a metro map, so in BioImage town. Again, everything CC BY, and you're welcome to use these. And in fact, we have, I guess we're starting a little working group to discuss the problems based on this metaphor. In trying to draw what I feel it actually looks like, it's more like this. So there's a very complex map. There are different colored routes where people have to, you know, they have to get off the train and get on a bus, and then after they get somewhere, they have to get off the bus and they have to get on a bike to finally get to, you know, to where they're going with their data. It's really quite complex, and I answer lots of questions from users about, you know, I've got data up here, how do I get it down here? And there's no straightforward answer at the moment. So what we do at OME is try to start from the ground up. I guess this is why Judith and the others put me in kind of the engine room of the car, is going, you know, someone has to start at the bottom and build up a common vocabulary or data model, as we call it. and then build file formats on top of that, and then libraries on top of those formats. And then finally, you can build a system, a data management system. In our case, it's called OMERO. There are others out there, but what's important is that we work together on as many of these components as possible. That's really what, at the end of the day, what makes things fair, is having more people working on fewer solutions. You know, if everyone comes up with their own, then simply it gets unmanageable. So I can tell you, at least for many workflows, OMERO would be a fine solution. So, you know, for various modalities that are producing large amounts of data on, you know, on a daily basis, you would be able to take the data that's coming off of the acquisition systems, at least for some 160 odd native file formats, use bioformats to load that into OMERO, and then you would be able to do your collaboration. You know, so it is one solution that's out there that you can access. And I guess briefly to show you what that would look like, you get a platform where you can collaborate on data. It's really the pre-publication part of sharing your data. That means you have a permission system that lets you define granular small groups and say some people can modify the data, some people can't modify the data. You can work together to add annotations to your data. I caveat, importantly, the OMERO and the OME world don't say what those annotations should be. It's open for interpretation. But as Caterina will show you, other communities can come along and specify what those annotations should look like. You can have something like a living figure. So rather than copying all the images out into Photoshop, for example, you actually have a figure that knows what data you used to create it. And you can come back a year later or 5 years later and figure out what you did and perform these modifications, for example, during the revision process, and create another figure. And there are many examples out there that you can look into and get a feel for how you should, or how you can do your research data management. But again, we don't have one way of doing it, since there's not a standards body that's currently saying, you know, all of us are going to work in the same way. So it is about exploring the solution space and figuring out how you're going to do your work. What I think is also, though, important to get across is that these solutions, minimally the ones that OME provides, but really I think any of them that are out there, open source or closed source, only fit certain problems, and you really need to be aware of where you're existing. And then there are these inflection points where using the solution gets harder and harder, and that probably takes stepping back and evaluating what it is you want to then use. The inflection points I think about most often, and again, I'm down in the engine, is, you know, the image analyst who's working locally on, you know, a single workstation. When they start to share that, it gets more difficult. So they start using remote storage somewhere. That eventually has its own problems as well. Maybe there's just not enough storage in a lab to deal with the data. So we start talking about centralized storage. You start talking to central IT. Maybe even you start building up a national repository for storing all of your images. And then there's this world of the cloud, which is kind of vague and much more complex. And I think we're all trying to get our head around exactly what that means. So when working locally, one of the first problems you'll run into is just the many different file formats that get produced in bioimaging. That's what bioformats is for, is to help you get all that data out. As the data starts getting larger, you know, you can put it into an OMERO, and we know of hundreds of people who are doing this, hundreds of groups. What we've done to try to work on centralized storage is provide the image data resource. It's not for everyone, so we take tens of submissions each year. That's obviously not going to scale for all the data, even of just the people in the room, much less across the entire world. There's also the BioImage archive, which is intended to take all published data, but I would argue eventually they're going to hit the limits. You know, if all of us send all of our published data to one server, things will eventually fall over. And so when it comes to this domain of very large data, very many data sets, that's where we started creating the next generation file format to be able to put things into the cloud. I'll talk about that for just a few slides, since that's really what I've been focused on for the last several years. I guess I clicked on the wrong screen. There we go. So the hope would be with the next generation file formats that we would finally have a common API that takes data from any of the modalities that we are all used to working with, convert them into this common representation, have a pyramidal data structure, so a multi-resolution setup, so you can do this Google map style zooming into your data, but the same representation of the data could be locally for the image analyst, or it could be uploaded somewhere to the cloud or downloaded by another user, and it doesn't have to keep getting converted over and over and over again. That's, the first thing is bulk download should be possible. Interactive access should be possible without the need to download the entire thing. A user should be able to click and immediately see something. And fundamentally, there should be the ability to have, large scale parallel processing for those who have those needs. And the hope is that then by working on something like that in common, we can take this kind of very complicated metro map and we could reduce certainly at least parts of it. We can take some representation and say, well, we want our users to be able to get from A to B without having to change from bus to bike to train, et cetera, and simplify their lives. If you want to try that out, a very quick demo, I would suggest for most people to start with this NGFF converter from Glencoe Software. It's a GUI tool. It's probably the easiest way to get started. Conversion's not overly exciting. You drag and drop your images onto the GUI. It generates something on disk. That'll obviously take longer if it's a much larger image. And then you have something that you can drag and drop onto a tool like Napari and see your data right away. So that's a fairly standard workflow. Where it becomes more interesting is you can take that same representation of the data, put it somewhere on a file server. You don't need OMERO, you don't need other services. And you then have a URL that you can share with your users. You can put it in a wiki, you can set it in an email, and immediately you can open terabyte scale data without needing to download everything. So it does feel like it's going to make people's lives simpler, and I look forward to feedback on that. I have to get past the YouTube videos. Okay, so this is what I'm focused on doing here in Germany. So as Judith mentioned, NFDI is the National Research Data Initiative. It's kind of a large-scale funding initiative that runs over the next 5 years here in Germany to link all research output. We're not quite sure how that's going to function, but certainly in the bioimaging space, we're responsible for defining the standardization of the bioimage data type. What is an image and how are we going to represent it? But just doing that in Germany really doesn't achieve what we want to achieve. We really need to work beyond national boundaries. And so interested to work with whoever would like really to make this happen. So a great place for many of these discussions is global bioimaging. So I'm part of the image data working group. Currently what we're focused on is a white paper to help bridge the needs of core facilities and the needs of IT. It feels very much like the two sides aren't speaking the same language at the moment. And certainly I would say central IT often doesn't understand what your needs are. So why is it that bioimaging is complex? And we really need something that we can give to them and say, okay, this is what we need. Can we now build it together? And so we're working on that. And if you're interested in getting involved, please get in touch. And finally, and this, as I mentioned, communication is super critical. If there's nothing else you remember from this, it's if you have problems, if you need to ask questions, probably the best place to do it is on ImageSC. It's a discourse-based forum with thousands of people who are regularly asking questions. One of the things that I think is quite interesting is the community came together and wrote a paper saying, this is where we're going to have our conversations. So if you need to convince anyone that this is the place to go, take a look at that DOI. There's even a category where you can introduce yourself and just say, hello, my name is. I think that would be a great output of the meeting if we had just even a handful of people get set up with ImageSC. I would love to see you there. These are all of the software projects that are represented there. And we really do try to help as much as possible. We love hearing feedback, even that something's not working. Let us know, let us know, let us know. Certainly for the OME team, we try to respond to everything within 24 hours. We will definitely do our best. It's It's so important, I can't even express it. So finishing up, I think I'm roughly on time. The 3 points that I'm really trying to get across. Bioimaging data is hard. I guess welcome to the club, welcome to BioImage Town. It's what we have to deal with. But importantly, there are solutions that are out there. Try them, ask questions about, does it fit for your needs, and let's work together to try to figure that out. But we'd love to have you involved to the extent that you want to get involved. So thanks to the funders, as always, but also to the wider OME consortium for all that's been built together over the many years. Thanks.
Thank you, Josh. That was an amazing talk. And it's interesting because you're bringing the same final conclusion that we've had this morning. So we're all on the same page. All right. So while Oliver is setting, allow me to introduce you to Caterina Strambio-De-Castillia. And now we're going to move on with our analogy. So Josh just presented about what's under the hood, what are the car systems. And now we're going to have Caterina tell us about the car dashboard. And really she focuses on interfacing between what's under the hood and what end users are really needing. So Caterina is an imaging scientist at University of Massachusetts Medical School. And she's a CZI imaging scientist, and she has had very important contribution to some of the FAIR component of science, which is bridging fish omics and regular omics to microscopy, which is already something that I'm impressed of. So, Caterina, this is yours.
Hello. It's going to be hard to follow such a great talk by Josh, but I'm going to stick with the analogy. So the engine is essential to drive the car, but without the dashboard, it would be very hard. So there is, and when we are dealing with the dashboard, we all need to know a little bit about what it looks like. So it has to be written in a language that has to be common. So, I mean, if we go to the new car and it's completely different, it's going to be hard for us. So that's why the idea of metadata, information, and using common languages. So the word standard, think of it as a language. A language, after all, is a standard of communication. So we're talking about the same thing. And some of the people that were here at the exchange of experience yesterday might have seen some of the slides already, so I'm sorry about that. But we're talking about here pre-publication research data management. I'm going to spend most of my time thinking about pre-publication research data management. Josh already told you about the pre-publication versus post-publication. The pre-publication, the research data management aspect is the stuff that you have to do every day, and you already do every day. You may do it inefficiently. You write on your notebooks. You then cannot find your notes. You have Excel spreadsheet all over the place. So we are already doing it. Here we're trying to think about the fact that, first of all, focus our attention on the fact that we are doing this anyway, and maybe think about together of ways of doing it more efficiently and in ways that can communicate with the engine. Okay, so if we would be driving a car and our wheel is not connected with the engine, we're not going anywhere. So that is keeping. Let's try to keep in mind this idea that that's why we need a common language. We need a way that is connected to the engine, and so we need something that we can understand, but also the machine can understand. So what type of info? So it's all about information. Information that we collect ourselves. We can get it directly from the instrument sometimes if the manufacturers help us do that. And then they have to be transmitted to the engine so that the engine can move them along. So, and how are we going to do it? Building bridges is a concept I'm going to concentrate on, and building community tools as bridges. So this is good for the fact that we need to trust our data. So in order to know trust our data, we need to know what it is, how it was done, so that we can reproduce it and we even interpret it. Then it's for organizing data, so we can find it. And then I pay attention to those boxes, they have labels. That's a point. So yes, you might have things very well organized in boxes, but without labels, you cannot find the content of the boxes. And all of this is about, again, the concept of FAIR. You've heard it already from Judith this morning, so I won't tell you much more about it. But think about the fact that it is good for science, but it's also now going to be mandated by more and more funding agencies. So it is important to do it also for that reason. It's good for scientists because our impact is much better if we produce good science. It is good for science in general. And in general, this is an idea of the cycle of the whole cycle of the data and keeping information about each aspect so that we can make data that is already fair from the start. And if we get to this point, oh, I cannot point. If you find, for example, if you get to the collection and you don't remember anything about what has happened before, okay. So yeah. So if you get to one point and you don't know anything about what happened before, you're going to be in trouble. So writing things down in an organized manner here in form of metadata, it's a good present also for yourself in the future when you get to point here. So metadata is, you can also think of it as a present for yourself. And then, of course, it's going to be a present for yourself where you're going to have to do the post-publication. So you're going to share because people now are mandating it. So then you're going to be very happy with yourself if everything is organized. And so the point is, as Josh told you, we don't have all the solutions. We are trying to develop together the solutions. It's essential to communicate with each other. But the idea that maybe you want to come out here from here is this is doable, it's possible, and we can start to think about it. And if we're starting to think about it, talking to our users about it, we're going to move forward. So here the idea is that there are different phases of the imaging experiment. Before the microscope, at the microscope, and after the microscope, we need to keep track of all this information. We have tools such as OMERO to do it. If you don't have OMERO, you can do it other ways. And then after all of this is set up and organized, when you get to have to share it, you're going to be in such a much better situation. So again, there is also incentives from the top down. So what kind of information are we talking about? We're talking about, again, metadata as the treasure map to get to the data. And these are, I'm talking a little bit vaguely here and on a high level, but then Caroline will tell you more about, and you heard something from Beth already about, actually, no, you didn't hear it. You heard it at some people that attended the EOE heard it. But anyway, metadata is useful at all steps. Even it's not just useful for sharing and reproducing, it's useful for yourself or when you have to do analysis. So you need to collect information about sample preparation, the acquisition, and the analysis. And all of this has to be connected so that we can keep track of all the data of the provenance. So think of this word, origin of the data, the history of the data. So we need to build bridges. And we need to build community tools. So this concept of, so we were using the car, now we're talking about bridges. So it's all about, and Josh was talking about a cart. So it's whatever helps you understand this whole thing and help each other understand it. One thing that is really important is that we are not trying to force people to do anything that is impossible to do. We are really all about making things can be usable, that there is a lot of customization and modularity in what needs to be done, so that there is clear added value, and a lot of it has to be automated as much as possible. So one of the things is that building bridges between data life cycle, the history of the data, so provenance, and the data, and this is done through metadata. So the metadata is used to connect the dots. And right now, the metadata, it's all over the place. So it can be found in multiple different places and is fragmented. And so we need to figure out a way of defragment it and organize it. So I mean, you heard from Judith this morning about the project that is happening in Canada, Bioimaging, in which, you know, the group, my group at UMass Medical School is heavily involved with. And the idea is that we are coming up with possible solutions to deal with each of these aspects. We are, for example, developing spreadsheets to capture the information about experimental and sample metadata. We are using tools such as MicCheck, MetaJ2, and Micrometa app that were developed to capture microscopy metadata based on the 4DN BINA OMI Quareb data model. I mean, you heard from Josh what a data model is a way of a language for representing data and metadata. And so these tools and this model you can find in the Nature Methods issue that was released in 2021 if you want more information. And then we also need to have metadata about the analysis and what we're doing, and that can be done by using tools such as Cell Profiler that actually allows you to keep track of the workflow. So different tools are used across, and then we need to have a glue to keep them all together. How am I doing with time? 1 minute? 6 minutes. Okay. And then, you know, again, the concept of community to get together to agree on each of these different aspects. I don't have time to tell you much about each individual solution, but come back to me, and if you come to the exchange of experience tomorrow, you'll learn more about it. One important thing is the common language, and RAMBI is what Judith already told you about, is a way of actually start giving you a map of the kind of information you need to capture for all the life cycle. So there are all these different bubbles that represent the areas that you have to make sure that you collect information about. If you go to RAMBI in this QR code on Nature Methods, the same issue where you find information about the other tools, you will see that there is an Excel spreadsheet with few fields that can at least give you an initial map of minimal information you need to capture about the sample preparation, the image acquisition, and the image analysis. What we're doing with Canada Bioimaging is developing Excel spreadsheets to capture this information based on RAMBI. And if you want to know about more about that and join this effort, and we can share what we have learned, join us at AIM, what you have heard from Judith about. And then the other bridge that I want to talk about is, to bring it all together, is the data to the research management infrastructure and tools. So again, this bridge, in this case, let's go back to the analogy of the dashboard. So now we have the data, and we have to connect with the engine that is going to allow us to do something with this data. So this is the same map that that Josh was telling you about, that Josh and others developed, the BioImage town map. But this is how it is for most people right now. It's fragmented. It's basically a series of subway stops with very limited connection between them. So this is really, and somehow all of these aspects, the experiments, the acquisition, the data, visualization, the analysis, have all to go in journals, which is already hard enough. A lot of times, even to do that, it's hard because we haven't taken sufficient notes. But now they're telling us that we have to share all of this. So obviously, that's where the idea of building a bioimage metadata, subway map that actually connects across the different subway stations is essential. So, and one example is the idea that we produce fishomics data. We have developed formats so that then it can be used for, these formats can be used by people that develop algorithms. And so the idea is that we need to connect all of these different areas. For example, here we need to import the data together with the experimental metadata, import the images together with the microscopy metadata, and we're using OMERO as a glue so that then we can connect all of these aspects, which is the analysis. And finally, all this part can be pushed to public archives. You have already heard about OMERO. How much time do I have? 2 minutes, okay. So what we have developed, you've heard a little bit this morning, is, so if you're interested in this, please let us know, is we're developing tools to make sure that information about the sample preparation and image acquisition metadata acquired with the tools that I can tell you more about. Excel spreadsheet, micrometa can be automatically imported into OMERO, and then they can be used for directly annotate the images so that you can find all this information with your images, and then this can be the start of the analysis pipelines that you can use, for example, using Cell Profiler, what you heard today. One final thing I want to tell you about is that where is all of this metadata that we're talking about? Here we have an image with some information in the file header, but then there is other metadata that we have to connect: the sample, the method that was used to to produce the image, the instrument, and then the analysis. So this is right now, a lot of it is fragmented, but we are all trying to build a systems so that all of this is connected. And when you have images, you know all of these aspects, so that then it can go in publication and in shared facilities. I think I'm out of time. 1 minute, okay. So to do that, we are actually, with Josh, we're developing a project to, one of the aspects is about, sorry, going back a second, about actually collecting the information about the instruments, so, and connecting it with a permanent identification of the instrument. I'm not going to be able to tell you much about it, but if you want to learn more about this persistent identification of instruments and how we can collect some of the metadata in that way, so that the image only needs, and the paper only need an ID to know exactly what instrument was used to produce it. Get in touch with me during the meeting, or if you are online, you can send me an email. This is a project we're doing in collaboration with ABRF, Core Marketplace, and RRID. Josh is part of, and Jim and Judith, who are all here, are part of this project. And the idea is to make sure that we have a persistent identification of the instrument and the persistent identification of the description of the instrument, so that all of these things can be connected together. Thank you for your attention.
Thank you, Caterina. That was fantastic. And you need to admit that when you take on such a task, be passionate about your work, and that's what you are. Get passionate. It's very refreshing. So we're moving on, and with our next speaker, and to again take the analogy we've chosen to have today, we heard about the car systems, we heard about the engine, the interface, the dashboard, how critical it is, and now we're going to have our driver. And Caroline Miller is going to be our driver. He is a histologist and imaging scientist with many years of experience. And she has a very interesting perspective for us today, because she's been working a lot with small and medium biopharmaceutical companies. And it will be very interesting to hear her take on what we've been doing to make her life easier in terms of imaging analysis. Thank you, Karen.
Thank you so much for setting up my presentation so well. It is really amazing that I can sort of wander my way in and just tell you about the sort of nitty-gritty details of an experiment from the ground level, but using all the principles that Josh and Caterina have already talked about. Imagine that my lab is perfectly set up when it comes to the Josh world. The engine is running and primed. Imagine my dashboard has all the measures that I need, and that I can see them and visualize them, and that all of that is working. And I'm going to walk you through an experiment all the way from tissue preparation through to image analysis, and sort of talk a little bit about the metadata that I would be collecting at each one of those stages. So first, I wanted to sort of introduce a little bit about the drug discovery process. And if I can find, where's the pointer? Is it there? Sorry, I can't see it. Is that? Ah, perfect. So in the drug discovery process, there are a large amount of different processes that occur, but you will find imaging in the sort of latter part of those processes. So for example, sorry, that's a little small, but it says functional imaging. And the functional imaging is more like electrophysiology or in vivo tumor imaging. We also have in vitro studies as well as ex vivo studies. These are often very specialized, often fluorescence studies with cultured ex vivo cells, reconstituted tissues, and we're using a wide variety of microscope techniques in that space, usually producing 3D data. What I'm going to talk to you about today is thin section analysis, so histology processing and whole slide imaging. These techniques are often used for both in vivo studies where we're looking at drug discovery, where we're taking the drug and putting it into the animal to see whether we're curing the disease we're looking for, as well as looking in human archival tissue to look at our drug targets, and also then looking in drug-treated humans for stage 1 and 2 clinical trials. So today I'm going to give you a little bit of an overview on all the points of histology. I'm going to hit the high points on these. I could keep going for hours and hours and hours, so I've definitely had to trim myself. And each one of these steps has important metadata. So I'm going to be talking about the metadata that I would collect at each of these levels. So we got to start at the beginning. Yeah, so we have a project, and then we have multiple experiments beneath that project. And then we all wrap everything back up to the project level to do the discussions and next steps and how we move forward with the projects to the next levels. An experiment can span multiple departments when it comes to a biopharma experiment. So we have the pharmacology and chemistry departments. They're producing the drugs that we're actually using to treat the animals, and then hopefully the humans later on. We have the in vivo team. They're the ones that look after the vivarium. They do the animal models. They perform the dosing experiments. They often do the necropsy as well. So that's the kind of link between me and the animals themselves. We have the histology and imaging team, where we perform the thin section analysis and the processing. And then we also have an in vitro team as well, which may require non-fixed tissue. Those guys can often put a little bit of a crux in the histology processing because they need something different out of the tissues to what we would normally do for histology. We want to be obtaining consistent and valid data and associated metadata. And this all starts with collaborations and good planning between each one of these teams. We should all be in sync. I know it doesn't always happen, but that's where we're going for. It's really important to have correlation of imaging data with other orthogonal experiments to really allow to have confidence in the results of the final project. You know, because we're going to be doing subsequent experiments. If you get it wrong way back over here, you're just going to be compounding your errors when it comes forward. So we want to make sure that we get all of the experiments. We want to make sure that all of the experiments within a project are matching up to the same results. And that takes good collaboration between these different teams. So now let's talk about the imaging experiment. So again, I say it again because it's really important that we have to have good communication and documentation between the different teams. Histology and image analysis is really specialized. You often don't find in biopharma that just like you will in academia, you don't have the same person doing the entire experiment. Large companies and academic institutions often have internal histology core facilities, but smaller companies utilize contract research organizations where they send out their histology for different portions of the analysis. Again, clear experimental design and documentation. You want to include all the stakeholders in the experiment. You want to have clear defined goals of what your scientific goal is and what your reporting goals are. Metadata really helps prevent surprises for the other project members. So if we look at this image right here, this was some brain tissue sections. And when we actually got to the slide, it was a bit of a surprise that we had the holes in here because the holes were taken for another type of analysis. So by the time you get to your slides, there should be no surprises. You should not see that. Okay, so for an imaging project initiation, you want to have project level information. So we want to know why we're doing this experiment. We want to know what our treatment cohort groups are. We want to know how you've determined the size of those cohorts so that we know that our statistical significance is good enough. What are the tissues of interest, and what are the target molecules and pathways? From an imaging level we want to know what are the endpoints? What do you want to see? Are we going to do qualitative analysis? Are we going to do quantitative analysis? What kind of molecules are we looking for? What kind of segmentation do you want to do? What kind of compartments do you want to segment your images into to then do further analysis and actually get more information from your images? Do you want what type of analysis? So we can have simple area measurements of staining positivity. We can look at cell type determination, the target staining expressors, and we can also do nuclear counts with positive and negative stainings. Also, it's really important is how are we going to report this out to the rest of the team? What kind of statistics are we going to use? What charting and what reporting tools? So the project level information, we always want to capture this in our metadata, again, in Josh's wonderful system that he's created for us. And the imaging and experiment information really allows the histology team to determine what's the best way for processing the tissue to get the best result out at the other end. So here's where I get on my soapbox a little bit. I don't know if that translates well, but it's one of my big points is that necropsy and tissue handling at the start of an experiment is vitally important to actually getting a lot of the other work done correctly. I really prefer perfusion fixation. It provides fast and even fixation throughout an entire organ. It also stops any of these kind of DAB false positives, and also in our fluorescence, the nasty autofluorescence of those pesky red blood cells in our images. I know that everybody knows what I'm talking about. It is a little harder to control archive human tissue fixation. And also you've got to be aware that some other downstream techniques might not like the whole animal to be fixed by formalin. So in that case, I still advocate for a PBS flush of the animal, and then we can do post-fixation. Post-fixation should always be done for each organ. It's going to depend on the type of organ. I'm not going to read through all these, but I'm happy to share my slides and also talk to you about it later on. So don't be afraid of providing a lot of information. So for example, this on the left is my perfusion protocol. This is kind of for a company I was working for that didn't quite work, but that's okay. I like the perfusion protocol that I have. And I will say that at this perfusion level is where you really want to be careful about the metadata that you are collecting, because this is the stuff that's going to mean that you can actually perform the rest of the analysis when it comes to pulling out the numbers and doing Excel analyses as well as performing, creating charts. So we want to know the animal and tissue ID numbers, right? That's important. We want to know which ones, which cohorts each one of those animals relate to. We want to know the protocols for perfusion, time of fixation, fixation type. All of these information about the types of fixative can really help figure out if there's any problems later on down the line, why do the problems happen, and can also help interpret the results as well. At this point, we also want to know where all the different tissues are going. So again, like I said, multiple different departments could be taking those tissues and doing different analyses. We want to know where they are so that everybody can then get a hold of the results when we come to the conclusion of the project. So another one of my little soapboxes is, again, right at the beginning of the process, is the dissection and the tissue grossing. So consistent and well-planned grossing is vital to reducing variability in results. Right? We already have enough biological variability throughout all of our experiments. So producing making well-produced slides that have consistent looking organs on them, pieces of tissue, is really important to providing a good base to getting that good result. You can use tools like these little brain slices or tissue slices to actually get consistent height on your different micro grossing dissections. And when it comes to our metadata, which you'll see always in green, we want to actually know what the dissection protocol is, and we want to know where the different tissue got distributed to. And then why is that important? Why am I talking about this? Okay, biology, often doesn't have huge variation, right? So here you can see we have slight variation. It's definitely statistically significant, but there isn't that much difference between the bottom of one group and the top of the other. We want to make sure that our tissue grossing will reduce, good tissue grossing will reduce the experimentally induced variability. So here's a good example. We have aortic root visualization. So this is my little diagram I give out to people when we're cutting up mouse hearts. So I like to see it sliced on this angle right here. If we put the top of the heart down, so it's really nice to give the people you're working with a nice flat surface to allow them just to know exactly where they plot the tissue down. If you do that correctly, you can actually section through, you can get 60 to 85 micron sections visualizing all 3 leaflets in the aortic valve on each section. There, again, then you can choose the sections you want to analyze, and we're analyzing apples to apples and not apples to oranges. Here's a bad example. So I like to see the base, the apex of the heart. I like to see these the papillary muscle attachments. I call them the nubbins. So I like to see that across my entire data set. This next image here, it's okay. Let's ignore the sectioning artifact. We're not going to look at that right now, but it is a little tilted towards the atria, and it's a little too high in the heart. And then we see things like this in our data sets, and I say to this, just no. Okay, no, 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 not okay. This is not what we want to see. You know, what this does here is it reduces our statistical significance for our experiment, and then that's one animal that has to be thrown out of our experiment, and this is not good. So no. Paraffin processing, I'm going to speed through this because I'm running out of time. But so often the paraffin processing is a very difficult technique. It requires expensive pieces of equipment. It also requires expertise in the people performing the experiment. And therefore, a lot of it gets farmed out to contract research organizations. So what you really need to be careful of is that you are assessing the third party that you're going with. You want to assess their proficiency. You want to make sure that you have accurate tissue manifests so you know what's coming and going. And you also want to develop a positive working relationship, because if you're nice to them, they'll be nice to you. Here we want to capture metadata on the paraffin processing protocols, embedding protocols, tissue sectioning. Also, you want to make sure that the CRO gives you detailed notes about any deviation from the protocol that you gave them, because then at that point, you want to be collecting that data. So if something goes wrong later on, you can correlate it to what happened at the CRO. Okay, so staining techniques. There are so many different types of staining techniques. There's no way I could go into them. We have H&Es and special stains. We have single-plex chromogenic or fluorescent immunohistochemistry. We have multiplex chromogenic or fluorescence, and we can also have in situ hybridization. So the histology team will help you choose which of these techniques is the best for visualizing your molecules of interest. I want to talk a little bit about immunohistochemistry auto-staining machines, because I don't feel this community has seen them as much. They're really fast and efficient, and they give a much better result, a cleaner result than manual staining. One of the other really great advantages is that the it is antibody species agnostic. So I can do mouse, mouse, rabbit, rabbit if I want when it comes to doing multiplexing, because of the way the machines work. The tyramide amplification detection systems that have come along when it also comes to the IHC staining machines have really made it much easier with difficult antibodies. There's much less going back to the as well as better antibodies as well from the companies. There's much less going back to cryo sections. The assays are easy to set up, and there's no more pat pens. There's no more jiggling your slides around in the oven or doing anything like that. What's also really nice is you put it on in wax, so the section still has wax on it. The machine will de-wax it, do the antigen retrieval and all the staining, and then you just have to mount the fluorescence slides later by yourself. So metadata at this level, we want to be getting the antigen retrieval steps, antigen catalog numbers, detection reagents, and then staining machines actually allow you to print out the protocol, so you can add that straight into Josh's lovely system. A little comment on, all right, okay, I'm gonna have to speed through this. I put too much. So IHC multiplex development really needs to be well controlled. And I can talk to people about how I would do that later on. I wanted to give you a quick example of a very well controlled multiplex analysis, a multiplex slide. You want to be seeing exactly the same in your fluorescence as what you see in your DAB, your initial bright field analysis. And the whole slide imaging has really sort of revolutionized thin section analysis. You don't have to pick multiple high power views anymore. So good tissue prep and imaging are really vital to actually producing the best result at the end of the day. There are multiple commercial slide scanners available on the market. Bright field and fluorescence, you can do 10, 20, and 40x. Some of them do have 60, but gets a little messy when you have the 100 slide capacity on it. You should really make sure that you're assessing the file format so that you can have compatibility with downstream analysis. So again, the metadata we want to collect here, we have microscope acquisition and hardware settings, file names, file locations, file formats, all nicely stored in the system that Josh has created for us with the schema that Caterina has laid forward. And I'll finish just with one image analysis example, where here we are taking a mouse heart, we are staining with Masson's trichrome, and then I'm doing a segmentation analysis to pull out the fibrosis in those hearts from 2 different cohorts. If we look here, the blue signal is the blue label here is our fibrosis label, and the orange is our muscle label. And then we have quantitative data that we can produce charts for, which will then come back, those experiments will come back to the team, and we can determine the result as a whole. Hello? Okay. And I have one more example, but I'm going to stop and say that metadata is also really important when it comes to the image analysis as well. We want to know the pre-processing steps, the analysis pipeline that we used. We want to know the post-processing steps, and we also want to know what our outputs are. From this, we can get our qualitative reports. We can also add to our qualitative reports, as well as the chart statistics and details. I'm happy to talk about any of this later on with people, and thank you very much for the opportunity to speak to you today. Okay.
All right, thank you very much, Caroline. So I hope we were able to display to you the value of integrating the car system, the dashboard, and a good driver in terms of managing and sharing data. So we're about to get started on the Q&A session, and I'm going on this side.
Hello, from the online audience, we have Claire DeSenza asking, thank you so much for this overview. Any tips on making compliance easy/natural on metadata collection, i.e. ensuring those in different teams collect the info they need to have all this metadata afterwards?
I mean, I think, you hear me, yeah? I think a lot of that comes with the system, with the engine, right, and the dashboards. You really have to have that basis underneath you that makes it easy for people to add their data to it. If you have to log in via a VPN and then go through 2 different login pages, then people are not going to put that data in, right? If you allow it to be front and center and work within their workflow, then you're much more likely to get the compliance of getting the data. I also say, like, do you want good results, right? Because if you want good results, then you have to give me the information. So there's definitely a learning process, I find, between the different teams, but you can get there eventually.
If I can add, what? Oh, Josh wants to? Okay, I'll wait for Josh. Okay, all right. So, so thank you, Josh. So yeah, so for example, while we're waiting for this perfect subway system, we can start doing something. For example, one thing that we can do through AIM is share these Excel spreadsheets in which you can at least start collecting this information based on the RMB and the NBOQ and the MBOQ standards, and then those can be kept next to your images. That would be like an initial way of doing it, even if you don't have OMERO or anything else, so that at least you keep that information next to your images and it's always going to be the same. Another way is if you have electronic lab notebooks, you can try to standardize things there for, again, using these examples that we can provide.
Yeah, I'll just add, I mean, there are definitely lots of pieces to the puzzle. I assume you can hear me, right? One of them is, of course, we have the hope that machines will start producing metadata for you, right? I mean, so it should capture everything and it should send it down the, I don't know what metaphor we're on anymore, but somehow the machine should do the work for us. That's the original acquisition systems. Something else that certainly some folks in Germany are working on, and I assume it's happening everywhere, is more use of barcodes and QR codes. Label everything, give everything a name, and then hopefully someone down the road, or before it gets to you, has added the metadata, and all you have to do is scan the barcode, right? Giving things a consistent, unique name is already a pretty good start to making things work right.
A question here. So it's a question related to metadata, and specifically to make it happen the FAIR principles all over the world, which might be through the publication ecosystem. I mean, I think that several people here, including myself, will love open source and just contributing to communities, but there are plenty parts of the international community that really believe in the peer review, and I also and in the editorial system. So I wonder about international politics to make the science to be reproducible in terms of metadata. Specifically speaking through the journals, through the funding agency. I know the case of the NIH, but I wonder if there is a global perspective.
Well, so QUAREP is actually a global initiative. So that for example, taking again the RMB map, there is that piece that talks about that is called image acquisition, and QUAREP is basically taking that piece and working with people literally from around the world, and we hope other people will join to flush that out, to develop those standards. I think there has to be both a bottom up, so there is going to be community getting together and doing it, and then there's going to have to be some top down, so that, as you're saying, different decision makers and the people that hold the purse, frankly, gonna be learning about this and actually start enforcing it. Some enforcement is already coming down the pipe. But I think one of the things we have recommended, for example, in this response to the request for information for NIH, is actually support for communities. We need to talk. So people are saying, oh, it's impossible to come up with a common standard. We can do translations, but even to do translation, you'll need to work together and talk to each other. So there is no shortcuts to people getting together and deciding. Things that Josh does, they need people to get together and deciding together. So, and those are, we need more funding for situations like this in which we can get together and discuss things. There is no way around it. So I think that's a key piece. I don't think there is a governmental global response, but there is definitely community global response. Global Bioimaging, QUAREP, all of these people we are all working to try to get together. And I think it's already having a huge impact. Josh, go ahead.
So I'm not sure if there are any publishers who are in the room, but if they are, interrupt me and you say your piece. But speaking for them, so I think the first time we tried to work with a publisher was 2012, give or take. The Journal of Cell Biology installed an OMERO instance and tried really hard to do all of this correctly. I can't remember how many articles actually came with data, but it was interesting to sit on their side of the fence. So I'm just thinking about the journals, because from the people in the engine room, we would have loved if they had required images to be uploaded for every article, but they had lots of very good reasons on why they couldn't do that. Ultimately, they have to get articles through the pipeline and not data. And it's still just too hard for them to get data through the pipeline. And so it's going to be, I think it's one of these things where, everyone says, well, if the funders do it, and the funders say, well, if the journals do it and the journals, we kind of keep pointing in a circle. So I agree completely with Caterina. It's kind of like we all have to do it at the same time, and just be aware of kind of each of our limitations. Something I can maybe add, so we can't convince the funders to spend their money differently, but something you can do is, and I don't think it's been mentioned yet, when you're purchasing microscopes, think about what it is you want to come out of the microscopes. So since you're paying money for something, you can help make part of the puzzle fall into place. So and it does follow the money. So to the extent that you can, or as a reviewer on a paper, ask for the data. Those are the things that you can do in your roles. But everyone's struggling with the same problem that we don't have the capacity, we don't have the time, we don't have the money. And so it's just going to take all of us constantly saying it's important to make it happen.
Thank you, Josh. I'm just going to add that Nature Publishing Group has been very supportive for the QuaRep and BINA initiative, as well as EMBL Journal, right? Just to name those two.
Okay, my question is, my question is actually a follow-up question to Josh's comment. So I'm Winfried, I'm working now for Bruker, which makes me a representative of the industry, but I'm since years thinking about data management and data sharing. And so what I'm struggling with what does it mean in practical terms to collaborate between industry and imaging community? So I'm a firm believer that we have to work together to make it happen. I'm a firm believer that it's extremely important, but I'm struggling with the implementation. So it's simple things like this. Yes, we all agree we should adhere to standards, but what standard? What file format should I implement in my software? Yes, of course, metadata, but what metadata ontology should we use? Yes, we should work with open source software, but should I support Napari? Should I support ImageJ? Should I support OL2? What third, fourth, fifth package are missing?
Yeah, for what it's worth, I struggle with the same thing. So from the point of view of OMERO, for example, how many different pieces of software should I work together with? How many different cluster computing solutions should I implement? You always have to stop somewhere. A possible solution is somehow, as a community, we take a step back and we say, okay, we're actually going to define the process, the governance, the voting body that makes the decisions, and someone will come in and just make it happen. I don't see us necessarily doing that, but if someone wants to have a discussion around what it would look like, I guess we could do it. I think the most important thing is so this is what the Metro map is all about. Pick something and invest in it. So, having two people try to work together on something is already a benefit. In talking, I won't name any names, but in talking to one company, so there's been a lot of goodwill and saying, yeah, we think this is a good idea. We think we can work together. But for me, what the big change was is they said, we will hire, or actually in October, someone will start, we are going to put someone on the project of doing X. And that's huge. We're not going to find the complete solution, but investing real time, to sit down and implement a solution, I don't think anyone's going to complain. If you pick any of the solutions we've talked about today, or certainly anything on Image SC, and you get involved, I think that's already a very good place for us to be. I can't ask for anything more than that.
If I would like to comment. So yeah, so for example, we are already in Quareb developing a microscopy metadata standard, and we are already working with manufacturers to actually go through the model and revise it with the manufacturers, so that, for example, we have done this with camera manufacturers, Nikon, Zeiss, Olympus, and Andor, and many others have come to the table and revised the model, so that we can, basically they can agree with the model with us, and then they can actually provide that information about provide that information because we have agreed about what information they need to provide. As far as the how to provide it, Quareb is also working with Josh about developing shared frameworks that could be incorporated in, for example, using JSON-LD and LinkML to be able to actually provide that information. So, for example, if a company would like to work with us to put resources to develop such a framework, which we have Josh and other people have pretty good idea of what kind of things we would want to start doing. That is definitely another place where a company could intervene and help. So, for example, if your company wants to work with us, you can join any one of the work group that is relevant for Bruker. Probably all of them are relevant. Join any one of them and help us develop them, revise them, for example, on light source or filters or anything else. So we are right now, we actually have a working model. We need more support to get it going because so far it's mostly done by volunteers. So, but we definitely have a way forward.
Online question from Elka. Caroline, do you agree that in academia, it is often the same person doing all the experimental steps, so it often seems less urgent to them to collect the metadata because they will, quote, they will remember what they did?
Well, what do I say about that? I think I do, in the academic model, you are going to have the same person doing those experiments, right? It's part of our training. I also think for your own sanity that you need to be able to collect those data. Also for figuring out the experimental results at the end, I like to blind myself to these things when I'm doing the analysis. So I don't look at the sample cohorts until I get to my analysis. And if I really don't know which sample belongs to which cohort, I'm really kind of not in good shape when I get to the end. My last point is in sharing and reproducibility, right? If we need to be doing these experiments in triplicate to make sure that the experiments are sound and that we're getting the same result every time. If you're not performing that experiment the same every time, then you can't expect to be getting the same results out. Also, when it comes to sharing data, which, by the way, guys, NIH policy, if you get money from the NIH, then you're going to have to share your data. And if you're giving crappy data out into the world, then that's not good as well. So whilst I hear what you say, even if it's in your own chicken scratch that's in your notebook, I think it's okay. The best of all worlds would be to have the system that Josh is creating and the schema that Caterina is involved with. That's the gold standard so that we have machine readable and human readable data. But at the very least, you should be collecting all of these data points in your notebook so that you can see them later.
I can also comment. I've been one of those people at the bench doing experiments and doing it from beginning to end. So I'm very much aware of the fact that yes, especially when you're young, you're thinking that you will remember everything. But I also, all of us, I am sure, have found themselves thinking, I should have write more things down. You often, especially with a lot of the complex experiments we're doing, we are writing papers 2 or 3 years after we did the experiment, at least for some of the experiment we started with. So, I mean, it is a really, Caroline told you a system in which it's a gift to the next person, but here it's a gift to yourself in the future.
Hey, so this is kind of a question for everyone. So I love open standards, I love open data, and I'm glad to hear that there are some microscope vendors who are on board with kind of opening up that side of the equation. But my users want to use closed source image processing software. And some of those tools are very much a walled garden where your images go in and it's really, really difficult to get the processing steps and the final results back out. Has there been any effort to reach out to the closed source processing tools to try and get them on board with these standards, either to read the OMERO format or to interface in some other way so that processing metadata can be better captured? Because I think that's still something that's unaddressed.
Yeah, absolutely. I cannot be more on your side. No, that I know of, there isn't, I think, but Judith says that she hasn't, she knows of somebody, but I think we do need to do the same kind of process. Maybe Work Group 12 in Quareb can start that process, or Bioimaging Informatics can start that process, but definitely that is a big piece of the puzzle because a lot of the processing tools are proprietary. So even if they don't want to, and I have a problem in general, even on algorithms that are not for reproducibility for science, even algorithms that are not made available, you really don't know what's happening to your data. So, but at least make some aspect transparent. I think we should start having those conversations. But Judith has some more.
Yes, David, that's a very excellent point. In the past, in my experience, it happened 3 times where I had questions about what was being done in this black box, and I contacted the manufacturers and I talked with them, and they were very collaborative. So maybe not like, I don't think they're going to go completely open, but if we have an issue, it's important to talk with them. The other thing is, especially what we're doing with Quareb, for example, we want to encourage people to use open source. But if we analyze the same data set with different software and we get the same result, that's also a validation for confirming that we're doing similar things. So there's still work to do, but they'll collaborate, I think.
So there is a big issue of reproducibility, which is software itself. It is not peer reviewed, and it's made by humans, and humans make mistakes. So I wonder if you can comment on that.
So, okay, this is much stronger than before. So Beth actually mentioned that a lot of us are involved in this paper that just came out on Nature Methods called, it's for actually starting to address these aspects. So standardization of software, I don't know, but at least transparency of what, so I mean, we need to have some way of measuring, how the software works, and definitely the ultimate is that you have to have similar results or the same results, but at the very least, the first step is to make it as transparent as possible so that you share the actual code. That's a gold standard, or at very least you share everything that you know about what you used. But obviously that is not, they may not be sufficient, but it's a better situation of where we are now. So it's a step forward. So they have all the different levels. So the top level is the ultimate gold standard is you share the code.
Josh, do you have anything?
Yeah, well, this sounds like a beer conversation. Again, I wish I was there. So let's see. You froze. There you go. I see laughing. That's good. So as someone who is a reviewer of software papers, I'll point out a couple of difficulties, because I would love to get to the point where, you know, what you're talking about was fixable. Much of what I spend my time doing when I review a software paper is actually try to just get it installed. So sharing the code's great, but there's a lot of overhead that makes it quite difficult. I think one of the things that's quite difficult in this domain is the fact that we are fundamentally encouraged to each build our own software package to get funding, to get a publication, to get recognition. And that means you have a lot of responsibility. If you take something like the bioformats package, so we've been developing it since 2005. It's never been properly funded. I don't know what's going on. People are hugging each other. Hopefully we're not crying or something. It takes an enormous amount of resources to properly test and run bioformats. So every day we test it multiple times on terabytes of data to prove that we are getting exactly what we think. There's a team that every morning goes through every issue that gets raised and reviews them. At some point, maybe I should just give a talk on that. It's a massive undertaking, and it would be impossible for most people who are starting out building their own project. So how do we go about fixing that? I mean, I think there needs to be an ecosystem where someone can contribute and get recognition for their contribution, right? So that it's not, I have to figure out how to install their software in order to review it. It's a much smaller burden of them being able to say, I've added this one component, I got involved in existing software. That's a good thing, right? But it's going to turn things on its head, or we're going to have to turn things on its head to get us to that point. So yeah, again, one of these things where there's lots that needs fixing before we can get there. Go for it, Caroline.
And just to add to that, I want to take it one more further step down the wormhole, because I've found, you know, in my world, in the histology world, I have 80 samples, let's say, in a data set. And I will touch every data set, and I will look at the segmentation, and I will look and I will assess it. You know, I'm not going to say 100%, but I want to be within my statistical limits for my entire data set. Now, what I've recently found is that I use a specific piece of commercial software, and I've been checking my segmentation, but then the outputs are actually a whole different thing as well. So it's not just the segmentation, it's how the software exports the outputs. So for example, if I have multiple ROIs, I found that my software, if a segmented object transverses 2 different ROIs, it will double count it, right? Which is just awful. It double counts the area and it double counts the count. So that's not a bug, it's a feature. So how do we also know what the features of these softwares are, so that we understand what data we're getting out the other side? So.
Yeah. So I think this idea that, I mean, everything that has been said goes back to what Claire has been saying. It's a change of culture. We really need to work at all levels. Just making it, for example, I think what Josh was saying, it would be good for him to say, I think it would be great for him to, at some point, to give a lecture about what it takes to keep the open microscopy environment system going. Because I think a lot of it is taken for granted, and it's amazing how much work and dedication these people are putting in. And they're not being, they're being recognized. We all love them, but I think they're not being recognized as much as they should, because they are not building bright new analysis pipelines. So I think we need to switch to a place in which there is incentive that you can collaborate to the same software. Maybe not everybody's going to use the same exact software, but reduce the number of software we can all work on, and we contribute a piece of code and we get huge recognition for it, as much as if we would have like published a new piece of software that nobody will ever use down the line. because it's not maintained. And
Can I still add something? Yeah, or not? It's just that, well, I'm the kind of reviewer that go actually and read the codes. And for me, it's always that you have to go into the mind of the people that is programming in mostly is a mess. So what I really believe that it is missing is a culture of actually programming for a reviewing process. Programming in the sense that somebody will actually read the code and has to be understandable. If we manage to put that into the education of programming, then we will be able to sort more easily these problems.
Thank you very much. That's an excellent comment. So we have to wrap up, and so we're going to say a big round of applause to our speaker before we say goodbye to them. Thank you very much. I encourage you to come and meet Caterina and Caroline if you want to discuss further. Thank you very much.